Well, thank you, Oliver. I would like to just start by saying that it's been a great day. I've enjoyed both the formal presentations by many of the students and also the informal interactions I've had. An amazing group of high school students, and I look forward, if I'm still alive in 20 years, to see how you have changed the world. And that's my expectation for all of you. Today I'm going to talk about not so much about hard science and about technical aspects of research, but about the importance of communicating our science to the non-scientific public. And so as shown in the next slide, I think we've, one of the things that has prompted me to do this is the very low rate of acceptance of the COVID-19 vaccines. Even though they were clearly life-saving, I think that governments did a poor job of rolling them out. I think we did too much of telling people what they needed to do and too little of listening to them and engaging in conversations. And you can see that the rate of acceptance of the vaccine was quite low in the US and a bit higher, but still leaving some room to be for improvement in the UK. Now, since that time of the pandemic, one might say things have not gotten any better, right? So if you can't see this cartoon from where you're sitting, the teacher is asking the class, can anyone tell me how long this era lasted? And the era is the era of science. And so I think we see over and over again that people distrust science, they distrust scientists, and they instead would prefer to listen to what like-minded people have to say on social media. The next slide talks about why is it so hard for scientists to explain their science to the general public. And I think the reason part of the reason at least, is jargon. This is the technical language that is not a bad thing. It's fantastic in the laboratory, right? I can go into my lab and I could say, you should just CRISPR that allele into the endogenous locus in HEC293 T cells. It would take me 3 solid days to explain that to at least the parents here, and even perhaps some of the high school students, right? But the student in my lab knows exactly what I'm talking about. So jargon is an incredibly useful shortcut for technical conversations, not just in science, but in finance and in plumbing, and in the plumber who comes to your house. Jargon is great, but it inhibits talking about our work to the general public. So one thing that I've done to try to make a difference, and I think it's made a tiny drop in the bucket difference, I must say. I don't think it's really changed the world at all, but you have a copy of it in your bag today, which is my book, The Catalyst, which is talking about RNA science, RNA research for the non-scientist public. And I think this is the kind of activity that we all, as scientists, need to be doing more of. And I would encourage you high school students, when you get to the point of being in college, being in graduate school, that you spend some time learning how to communicate science to the non-scientific public, and then go out and do it. So the topic then of my book is RNA. And why RNA? Ribonucleic acid. Because I think that the 21st century has already been and will continue to be the age of RNA. DNA is old stuff. It's 20th century stuff. It's still important. It's really pretty boring, right? All DNA does is store information. It doesn't do anything. It's sort of like having a computer disk without any way to read out the materials, right? All of the action of the DNA is carried out by RNA and protein. And so that's what scientists are working on now. They're still looking at the DNA, but they're looking at it because they're interested in these downstream actors like the RNA and the protein. So as shown, when I give these talks to the non-scientist public, I see that people are generally comfortable with the double helix of DNA, because it's been around since 1950, since the Cambridge years of Watson and Crick. But when you talk about RNA, it's just a single-stranded version of the same information as in the double helix of DNA. But still people say, oh, that's much too complicated, right? Don't know about ribonucleic acid. But you can tell by the name, ribonucleic versus deoxyribonucleic acid, that they are chemically very similar molecules, and in terms of information content, they are more or less identical. The A, G, C, and T of DNA are copied into the A, G, C, and U of RNA, where U is informationally equivalent to the T of DNA. So one thing that makes this conversation a little easier is that now with the mRNA vaccines, most people have at least heard about mRNA. They know that it can give our immune system a heads up in terms of fighting a virus, and the virus that is, of course, still on our minds is the SARS-CoV-2 virus. So I think that this conversation with the general public about these mRNA vaccines sort of went sour when you started reading that the mRNA is a medicine. mRNA is a drug. And that makes people suspicious. It makes them apprehensive, because you shouldn't be taking drugs or medicine unless you really need it, right? And if you're not sick, it's hard to convince people that they should be taking a medicine. But I think that instead of starting out by saying that mRNA is a drug, it would have been better to say that mRNA is natural. mRNA is in every cell in our body and in every other living being on the planet Earth. And not only that, but it's in all the food that we eat every day, independent of whether you're a meat eater or a vegetarian, you're eating messenger RNA all the time. Not only that, but it's something that we've known how it's worked for the last 50 years, 60 years more accurately, because we've known that the DNA code is copied verbatim into the single-stranded RNA, which then instructs the cell's protein-making machinery in which protein to build. So groups of 3 informational units, 3 A's, G's, C's, or U's on the messenger RNA, instruct the cell to put a particular amino acid on the end of a growing protein chain. So it's not a newfangled invention. It's not something that was just found yesterday. It's something that we've understood the intimate workings of for many decades. So when it came to then using this information to make the SARS-CoV-2 messenger RNA vaccine, you might have thought that you could follow the pathway that nature takes, which is to make the gene, copy it into mRNA, and then use that to make the protein that would alert the immune system of what the virus looks like. It has to be a protein that's on the outer coat of the virus, because that's the first thing that the cell would encounter when a virus infects it. But it turned out that the clever folks at Moderna in Cambridge, Massachusetts, and BioNTech in Mainz, Germany, found was that they could instead just take a shortcut and inject the messenger RNA into your arm and let the human body do the heavy lifting of converting that mRNA code into the protein. So this was the first time that this old idea of an mRNA vaccine was actually shown to work. Now, these vaccines were not perfect, and I think a lot of the naysayers about vaccination harp on these features. First of all, they say, well, I got sick again. I got COVID 3 times and I was vaccinated, so it's not a very good vaccine. Well, but you didn't die, right? And it actually protects quite well against hospitalization and death. It's hard to know exactly how many lives were saved even in the first year of the mRNA vaccines, but the best estimates are somewhere between 10 and 20 million people didn't die because they took the vaccine. So that's certainly something to be very proud of and very excited about. They also say, well, you know, the virus is mutating all the time and then it escapes the immune system, and that's why the vaccine doesn't work very well anymore. Well, that's not the vaccine's fault. That's the virus that is mutating. You can't expect and I'll get back to this in a minute, you can't expect a vaccine against one form of the virus to be as good against another form. And finally, the cold storage, ultra-cold storage that was required for these vaccines certainly inhibited its broad distribution, especially to countries that don't have access to ultra-cold freezers. But, you know, that's, again, something scientists are working on. That is a shortcoming of these vaccines, but we're making progress on fixing that. So, on the other hand, there are some major advantages of these mRNA vaccines, and one is that they're so quick to make. Once you know that the virus has mutated and you know the sequence of the mutant form, it takes only a day to design a new vaccine that will now be specific for that mutated virus. This is not true of the traditional vaccine technology shown on the bottom, which is injecting chicken eggs. About 100 million chicken eggs are used just in the US alone, and hundreds of millions worldwide to make the seasonal flu vaccine. And it takes so long to grow up the debilitated form of the flu vaccine in these chicken eggs that the companies have to decide which strain of flu is going to be dominant in the next flu season before they know. And usually they get it only partially correct. And so that's why the flu vaccine is typically between 40 and 60% efficacious. Not, you know, still worth taking, but 40 to 60% isn't that good. So on the next slide, we see that, you know, one of the things we think the future holds for these mRNA vaccines, they can be produced so quickly once you know which virus you want to target, that we should be able to wait until we know what strain of the flu is going to be dominant in the next flu season, and then quickly make a vaccine aimed at that flu virus. So that is in progress. The idea of a personalized cancer vaccine is a bit of a wild idea, but it's actually working in clinical trials. And I can't promise you that this is going to be something that doctors will be prescribing in a few years, but for the moment, the clinical trials appear very promising for melanoma, and there's no reason to think that melanoma would be the only cancer to which one could become vaccinated against. And finally, there are about 3,000 rare genetic diseases that the cause is known, but we haven't been able to do anything about it. And although each of these diseases by itself may affect only a few hundred or a few thousand people worldwide, if you multiply that by 3,000 different rare diseases, there's actually millions of people who are affected by these diseases. And in the past, we weren't able to know how to intervene if we understood the genetic basis. Now with this mRNA technology, we may be able to give the person the healthy version of their mRNA and therefore of the missing protein and see if we can develop therapies around that idea. So on the next slide, we say, okay, if that's all that RNA could do, be a message, that's pretty cool. And maybe that alone would justify saying that the 21st century is the age of RNA. But I have some time left, and that's not all that RNA can do. So on the next slide, we see that there are many non-coding RNAs. It means they don't even care about this wonderful table of the genetic code. They simply are doing something that doesn't require them to be a message for making a protein. And there are so many examples of this, and so many examples yet to be discovered, that there are conferences in which people spend a whole week just talking about these non-coding RNAs. But I don't have a whole week with you. And so I'm going to just give 3 examples. One is that non-coding RNAs can have catalytic power. We call these ribozymes, where ribo stands for ribonucleic acid, and zyme is enzymatic activity. Secondly, RNA can bestow immortality. Whoa, that's a big one. But before you get too excited, it's only at the cellular level, not at the level of the human being. And then finally, RNA can control evolution. And this is the CRISPR gene editing revolution. So let's start with the first one. RNA can be a catalyst or ribozyme. And 2 slides over, Oliver. Thank you. And so when I first moved from MIT to Boulder, Colorado to start my own laboratory, I started studying pond scum. Now, why would any self-respecting scientist want to study pond scum? Well, it wasn't just any old pond scum, but it was instead this organism Tetrahymena thermophila that had this remarkable feature of having 10,000 copies of a particular gene. And if you're a biochemist, you want a lot of material to work with. And 10,000 copies of a gene is a lot, because most of you have how many copies of each gene? Come on, we don't have much time. 2, right? 1 from mom and 1 from dad. And so it's part of being a diploid organism. So 10,000 is a big deal. And what we found early on was that the RNA made from this gene was undergoing a cutting and pasting reaction called RNA splicing. Its internal structure was being rearranged. We didn't discover this phenomenon. It was discovered by Phil Sharp at MIT and Rich Roberts and colleagues at Cold Spring Harbor Lab in the US, and they got the Nobel Prize for this because it was a remarkable finding that, for example, most human genes have 10 of these interruptions that have to be spliced out at the RNA level. But what nobody knew at the time was the mechanism. How does this occur? How can the RNA be very accurately cut and pasted together? And here we had a system where 10,000 genes were pumping out RNA that had to undergo this process. So we thought we could identify the protein enzyme that was catalyzing this reaction. So we had a problem, though, because although there had to be an enzyme, and as James Sumner had said in his Nobel speech in 1946, all enzymes are proteins, our RNA seemed to be able to undergo this splicing reaction without adding any proteins. We put it through all kinds of purification, we boiled it, we added detergent, all kinds of things that are deleterious for protein function. And so if there had been any contaminating protein in our preparations, it should have been destroyed, but yet the RNA kept on splicing. So how is it possible to reconcile the apparent lack of a protein requirement for this splicing with the fact that all enzymes are proteins? And as shown on the next slide, this led to my graduate student, Paula Grabowski, giving me this as a Christmas gift when she was in the middle of these experiments. So fortunately, we were able to do something more scientific than picking the petals off the daisy to resolve this question, and we were able to show that the RNA by itself was folding up into a complex origami shape that allowed it to catalyze its own splicing reaction. And as shown on the next slide, this excited a group of researchers who were working not on Campbell's primordial soup, but on the events that took place on the primitive Earth almost 4 billion years ago, when the Earth had first cooled down enough and the intense comet bombardment had subsided enough for there to be a chance for organic matter to accumulate. And in a short time after that happened, it may have been 100 million years, but in a astrophysics sense, that's a short period of time, there was already primitive life forms on the planet. And when scientists tried to think about how that was possible, they came up with the world's sort of most daunting chicken and egg problem. So which came first, the information to pass down to the next generation, which in our cells is DNA, or the machinery, the enzymatic machinery that would copy that DNA into more copies, which in our cells is always a protein. DNA or the protein, well, they'd have to come together, but now there's a potential simplifying scenario. Because if RNA can be both an informational molecule, which it clearly is, in fact, the SARS-CoV-2 virus doesn't even mess with DNA, it's an RNA virus. RNA is plenty good for being a genome. So if the RNA can both be an informational molecule and a biocatalyst, maybe the RNA was both the photocopying machine and the thing that was being photocopied, and this would be a tremendous simplification for origin of life theories. So next slide. We're now, oh, and yes, should say that this led to the Nobel Prize in Chemistry in 1989, which I shared with Sidney Altman of Yale University, who found a very different kind of catalytic RNA in his laboratory. Now, on the next slide, we come up with the next example of non-coding RNA doing something really super cool. And this is telomerase, which, shown on the next slide, I have to introduce the ends of chromosomes, the telomeres, because this is what telomerase works on. Now, telomere, the telos in Greek means end, and the mere is something like stuff. So this is the stuff at the end of all of our chromosomes, and you can see in this picture of human metaphase chromosomes dividing that there is some DNA that is being decorated here that is common to all of the telomeres on all of the human chromosomes. And these ends are vulnerable. They need protection, much as the ends of our shoelaces need protection from unraveling. What are those little plastic tips called, after all? Aglets, thank you. The aglets are the protective, thank you, Oliver. You're also doing a good job changing the slides. Okay, so, but you know, the telomeres don't have little plastic tips, but they do need to be protected because, as shown on the next slide, if they're not extended each time the cell divides, then the telomeric DNA shrinks a bit each time the cell divides. And that's a good thing because it eventually causes the cell to stop dividing, which once you're an adult, you don't want all of your cells to keep doubling every hour, right? Homeostasis is a good thing. You don't, so therefore, they don't need a way of maintaining the chromosome ends. But there are some cells that do need to keep dividing, and these are our stem cells. And so and there were some talks by the students on the stem cells. And so you know that they can both, they give rise to two cells, one of which is another stem cell, which leads to self-renewal, but then also the other division product can differentiate into a more specialized cell. And therefore, the stem cells do need to keep dividing. And how do they achieve the protection of their ends? As shown on the next slide, they use telomerase, which of course, because this is an RNA talk, has an essential RNA component which collaborates with a protein called telomerase reverse transcriptase, which we discovered in my laboratory in Colorado. And the combination of the RNA and the protein gives rise to this complex that puts extra telomeric DNA on the ends of the chromosomes before the cell divides. So it maintains the ends and allows continued proliferation. Now, the idea that this could be a fountain of youth, this telomerase, has led to many people trying to lengthen their telomeres on Amazon.com. And there are lots of nutraceuticals that are sold that claim to be clinically proven to lengthen telomeres and to activate telomerase. But the truth is that they have not been through what we would call a clinical trial. They are allowed to be sold because they are harmless food supplements, and I think they're probably so harmless that I doubt that they actually extend your telomeres. I'm not sure. It is possible, but if they did, would that necessarily be a good thing? Well, there's a dark side to this immortality, because there's a kind of cell that is immortal, that is the scourge of especially older adults, and this is tumor cells, right? They can divide indefinitely, and they do this by reactivating the telomerase by upregulating this protein that we discovered, this telomerase reverse transcriptase. How cancer cells do this was unknown for many years, but then Levi Garraway at Harvard Medical School and his student Franklin Wong, and also a group at the German Cancer Center in Heidelberg, figured out that there was a mutation in the control region of the gene, not in the part that makes the messenger RNA, but preceding this in the regulatory region. And this single base pair mutation was found at a phenomenally high level in multiple cancers. And I still find this to be such a remarkable story about evolution, because 80% of the melanoma tumors around the world are able to continue dividing over and over again because they've upregulated telomerase through this mutation in the regulatory region. Think about it. There are 300,000 new cases of melanoma around the world each year, and if you sequence the tumor of those patients, you find that 80% of them have upregulated telomerase by making this mutation independently of the others, right? And you sequence the normal tissue next to the tumor, and it does not have this mutation. So it's a new mutation that is driving cancer. Many other cancers, glioblastoma, bladder cancer, liver cancer, also rely to a large extent on this promoter mutation. And interestingly, other common cancers like breast cancer and colon cancer still upregulate telomerase, but they don't do it through this mutation. They use an epigenetic mechanism. Now, the next slide, I think, brings us to the last kind of non-coding RNA, and this is the one that gives CRISPR genome editing its enormous specificity. Next slide shows what gene editing really is. If you think about all of the information in all of the human chromosomes as a Word document, a text file, then it would take about 1 million pages in a normal size book with a normal size font to write out the whole human genome sequence. 1 million pages of text. Yet CRISPR is able to find within that 1 million pages a particular sentence and a particular word within that sentence. And then that can be used to edit mistakes in the text, as I've shown here by the letter that is in red. Okay, so pretty remarkable search and find and replace mechanism to work on this scale in the microscopic environment of our cells. Now, how does this work? Well, or who did this? Well, the Nobel Prize was given to Emmanuel Charpentier, Jennifer Doudna, just 5 years ago for their discovery of CRISPR genome editing. So clearly this was considered to be a huge landmark in biology. And we were particularly excited about this Nobel Prize in Colorado because Jennifer got her postdoctoral training in my laboratory. And here she is at a laboratory reunion along with some other scholars who are also professors at universities around the world. Now the next slide shows sort of simplified version. Again, remember, this is for the general public, the non-scientists. So the little red scissors is a way of simplifying what the CRISPR does. It cuts the DNA double helix. But it isn't really quite this simple, right? So shown on the next slide is how RNA, shown here in pink, guides the CRISPR machinery to find the spot on the human chromosome that's going to be cut. And it only locks down on the DNA when 20 of the informational units, the A, G, C's, and U's, on the RNA match the corresponding sequence on the DNA. And then the RNA pairs with the DNA, holds the CRISPR machinery in that spot, and enzymatic activity that's provided by an associated protein called Cas9 then cuts both strands of the DNA. Now, why would cutting DNA be of much interest? I mean, that sounds like a really bad thing to do, to damage the DNA. But it's such a bad thing to do that all biological systems have, through the eons, built up repair machineries to repair cut DNA because it is so deleterious to have your genome severed, right? And this can happen, for example, by radiation or by chemical damage or by multiple means. So on the next slide, we see what actually happens after the DNA is cut is that the DNA is repaired, not by the CRISPR machinery, but by machinery that's already present in all cells. And it's during this repair activity that one can introduce new sequences shown in green into the DNA. And these can, for example, repair a mutation that is a deleterious mutation, or you can add even a whole new unit to the gene, which then will be transcribed into RNA, which then will be made into a protein. So you can append, for example, a fluorescent tag onto your favorite protein so that you can follow its motion around cells. So shown on the next slide then, there are also, this is so powerful, and many people could do this in their garage or in their kitchen at home, right? It's that robust of a technology that one has to start worrying about some of the negative things that could be done with this technology. Just to cite a few of the title pages of books that have been written about CRISPR in the last few years, it's been credited with the unthinkable power to control evolution, the awesome prospect of editing humanity, the chance to direct the future of the human race. So this is just like much of the science that you have talked about yesterday and today can be used either for good or for evil. And so I think it's important then that we as scientists have the responsibility not to tell the non-scientist public what they should be thinking, but to engage with them in conversations, to listen to them about what are their concerns about this technology, and then to think about ways that we can prevent some of the ill effects that might be possible with this technology. So, though let me close with saying some of the great things that can be done with CRISPR. So as I've already said, or if I didn't say it, I should say it, it's already revolutionized what we do in the laboratory. The undergraduates in my laboratory are routinely doing CRISPR genome editing. It's that robust and that powerful. Medically, it might be able to be used to delete bad genes, but there aren't that many genes in our cells that we really would like to just get rid of. What we'd really like to do is on the bottom here, which is to repair faulty genes, to either fix a mutation. And we know the genetic cause of so many diseases. First of all, cancer has genetic mutations that are driving the cancer, one of which I already mentioned. Secondly, we've got diseases like sickle cell anemia, muscular dystrophy, cystic fibrosis, where the genetic cause has been known for a long time, but we haven't been able to do anything about it. And so now with CRISPR, there's a possibility, and in fact, there's already an approved therapy for sickle cell anemia that allows one to now edit the DNA in a way that can ameliorate these diseases. So on the last slide, or we're getting near the end, just in summary, I've talked about the fact that RNA can be a messenger, which is a fabulous activity, but also there are activities of non-coding RNAs that don't depend on the genetic code at all. RNA can be a biocatalyst. It can bestow immortality at the cellular level as a key component of telomerase. It can control evolution as a guide RNA that gives CRISPR genome editing its exquisite specificity. And then we see on the next slide that this, I think, justifies calling the 21st century the age of RNA. And lastly, my laboratory thanks you for your attention.